So we're going to talk about a very important class of receptors called receptor tyrosine kinases, or RTKs. This family of receptors is very important for growth factors. These are molecules that end in GF, like IGF, PDGF, EGF, etc. And also the hormone insulin, the insulin receptor, is an RTK. So let's start with the ligand binding from the outside of the cell. After all, these are called extracellular receptors for that reason. And what happens when the ligand binds to the receptor, the intracellular part of the receptor has these tyrosine residues. Remember, tyrosine abbreviation is Y. And the receptor itself has tyrosine kinase activity. So remember, we talked about protein kinase A and protein kinase C in other videos on GS and GQ pathways. Those are serine threonine kinases. They phosphorylate hydroxyl groups of those amino acids. In this case, the tyrosine has a hydroxyl group, and the amino acids that are on the intracellular part of the receptor, those actually get phosphorylated in an autophosphorylation type mechanism. So we're going to just depict that as something like this. And then we're just going to put those phosphates there on those tyrosine residues. Now, this pathway is pretty complicated. There's a lot of steps, and I'm not going to go through the exhaustive individual steps that exist. I'm going to simplify this and just start with the right side. We're going to have a protein that's going to bind to those phosphorylated tyrosines. It looks something like this, okay? And this is called PI3 kinase, okay? And the reason why it binds to these phosphorylated tyrosines is it has special domains, these little pockets that bind to those phosphates called SH2 domains, Stark homology 2 domains, and that helps get this PI3 kinase right up near the membrane. And the reason why it's called PI3 kinase is it's going to phosphorylate this phospholipid here called PIP2, and it's going to phosphorylate it at the 3 position in the inositol head group here. And so what we can do is we can draw like an arrow to indicate that this phospholipid PIP2 is going to get this extra phosphate right here. And now what we call this is PIP3, which is going to be named for the 3 phosphates that are on the inositol. So this is phosphatidylinositol 3, 4, 5 trisphosphate. Now, the reason why this is significant is that the 3 phosphate groups on this inositol serve as another anchor for other proteins that like to bind the PIP3. And we call these proteins that have these domains this plextrin homology domain, or pH domains. And just to keep things simple, we're just going to draw like a protein that looks like this. And we're just going to say there are various kinases that can go and assemble and bind up against these pH domains. Now, these kinases can then phosphorylate proteins, okay, just like we've seen before. So here's a protein. It's either active or inactive, and it's going to now get a phosphate, and now it can either be inactive or active depending on the protein. And what I'll point out here is that this is a kind of a complicated pathway. This PI3 kinase pathway is often it's referred to. This can do a variety of different things that I'm just going to simplify here and say that it could be involved in things like cell survival. So this is a way that the cell can increase its survival. Or there's also metabolic effects that promote growth as well. And if we're talking about the hormone insulin, its metabolic effects that it's most well known for are thought to be mediated primarily by this PI3 kinase part of this pathway. So that is really all we're going to say about this part of the RTK pathway. Now, on the left side here, we're going to show that there's other possibilities for the RTK pathway, and that's going to be, again, these proteins that can bind to these phosphorylated tyrosines, okay? And we'll just say, for example, this one is called GRAB2. GRAB2 is an abbreviation for growth factor binding protein 2. It's an adapter protein that can just bind and assemble up against the membrane. And then what happens is that GRAB2 can then attach to another protein. It could be attracting another protein. We'll just call that SOS, which stands for son of sevenless. And SOS is actually a GEF. It's a guanine nucleotide exchange factor, much like the G protein coupled receptor that functions that way too, changing the affinity for a G protein for GDP for GTP. There's going to be in this case, a G protein, but it's not going to be a heterotrimeric G protein. It's going to be a monomeric G protein called RAS. And RAS is going to be sitting here bound to GDP in the inactivated state. And what the SOS is going to do is it's basically going to make the RAS bound to GTP. And that's what this GEF is going to do. And that RAS that's now been activated is going to function to turn on a whole signal transduction pathway, which we are going to just illustrate with a series of arrows. Now, this part of the pathway is called the mitogen activated protein kinase pathway. Mitogen is a name for a growth factor. So we said that this is what growth factors use. They use this type of signal transduction. And so we abbreviate the mitogen activated protein kinase as MAPK, the MAP kinase pathway. And the way this works is that you're going to have a series of kinases that are going to be triggered here. And so we're going to start with MAP, again, that's mitogen activated protein kinase, but we're going to add two more Ks on here. It's a kinase. And another name for MAP kinase is RAF. That's an interchangeable term. And what the MAP kinase is going to do is it's going to phosphorylate MAP kinase. And it's going to put a phosphate on it. And MAP kinase, another name for it is MEK. So we have RAF is going to be phosphorylating and activating MEK, or MAP kinase, and then MAP kinase is going to phosphorylate MAP kinase, get a phosphate on it, and MAP kinase, the other name for it is ERK. So MEK is going to phosphorylate and activate ERK, or MAP kinase, and MAP kinase then, like other protein kinases, can take a protein, whatever that protein is, could be in the active state or inactive state, phosphorylate it, and now you can have less activity or more activity. And the MAP kinase can also go into the nucleus on a gene and can phosphorylate a protein that's sitting in the nucleus, and that could then affect gene transcription. In any case, these effects here are going to be involved in cell proliferation, and that's why we were saying earlier that we're talking about growth factors that are going to be enhancing cell survival, enhancing cell proliferation, turning on metabolic effects that promote cellular growth. And while we have these pathways drawn out here separately, I want to point out that there's a lot of crosstalk between them that's not shown here, where these are going to be working synergistically depending on what the particular cell is and what the particular signal is. Now, what about turning the pathway off? We have to think about that. And the way that the pathway is going to turn off would be done in a similar fashion that we discussed in other videos. We can have the ligand can unbind and just diffuse away. That's always going to be true. We also have RAS GTPase activity, right, that can go and reform the monomeric RAS G protein in the inactivated state bound to GDP. We also can have protein tyrosine phosphatases that can remove phosphates from phosphorylated tyrosines. So we can have that shown here, sort of tyrosine protein phosphatases. We can call that number 3. We also have protein phosphatases that can remove the phosphate that was put on over here, right? So we have protein phosphatases that can take those phosphates off. We call that number 4. In addition to that, we also can have lipid phosphatases. We can have a phosphatase that removes the phosphate from PIP3, and that would be something called P10. That stands for phosphatase tensin homolog. It's basically a lipid phosphatase, like I was describing here, taking off that phosphate off PIP3. We can call that number 5. And then we also have the potential for downregulation of the receptors if there's long-term stimulation, which we're not going to show here. That is also possible as well, chronic stimulation of the receptor. So these are several mechanisms of turning off the RTK pathway. And as you could imagine, if any of these mechanisms are not working right, you can have prolonged and exaggerated effects of these pathways. And I'll just mention that when you have a RAS GTPase mutation that's preventing this mechanism here, that can be a very common cause of cancers where you have this pathway continually on.